Welcome dear viewers to the video on partition coefficient, that is also referred as log P or log D, and the drug absorption. So the video is regarding the log P, its effect onto drug absorption, drug distribution, drug elimination, and also log P is a important physicochemical parameter while developing the drug molecule, and also it is to be considered while developing the specific dosage form. As per the other physicochemical properties like pK, solubility, particle size, log P is one of the key parameters. Log P gives you the measure of molecules lipophilic and hydrophilic nature. Log P affects the drug absorption, drug distribution, and drug elimination. Whenever the new chemical compounds are synthesized, the log P is considered so that it can be understood from the log P about the drug's absorption, distribution, elimination, and the route of administration. Also, log P is important physicochemical parameter to decide the specific dosage form, whether it will be oral formulation or sublingual formulation or topical formulation, or what will be the future of the drug molecule, that will be based onto the log P. Log P is the partition coefficient. It gives you the idea about hydrophilic and lipophilic nature of the drug molecule. The drug molecule differentially dissolve into lipid or water, and that is the base for its log P. Partition coefficient, that is log P, is the distribution or solubility of the molecule into organic or aqueous phase. Organic phase is used for oil part. Also, you can take oil and water. How much drug is dissolved in the organic phase and how much is dissolved in the aqueous phase is measured by the log P. Log P, that is partition coefficient. Whenever the log P is considered, that time you should understand about the definition. Log P is the physicochemical property of the drug molecule. Log P may be defined as the logarithm base 10 of the partition coefficient, that is P, denoted as P, which is the ratio of the compound's concentration in the organic phase to its concentration in the aqueous phase. So basically it is the ratio, and it is denoted by and calculated by logarithm to the base 10 of that ratio. Now, what is the effect of log P and absorption? See, the membrane barriers are made up of the lipid bilayer. You might have read about the lipid bilayer membranes. So, whenever the absorption is there through these membranes, the molecule partition into the lipid membrane, and then it partition into the aqueous phase from the lipid membrane. That means lipid membrane is the barrier, or it is the layer between the aqueous phases. GI absorption membrane is made of the lipid material and it is hydrophobic in nature. So the drug is required to be having some lipophilic properties to get partition in the lipid membrane and then hydrophilic properties to cross the lipid membrane and enter the aqueous phase or hydrophilic phase. Like blood plasma is aqueous phase. Lipid membrane and blood are the two immiscible layers. Again, the GIT membrane and blood-brain barrier are the lipid in nature. Log B not only affects the drug absorption, but it also affects distribution and excretion or elimination of the drug from the body. That's why the log P is considered in the drug design, drug development and formulation development. High log P value of the drug indicate that the drug is having high lipophilicity and that's why it will impact onto the bioavailability of the drug. Also, high lipophilicity will have impact onto drug distribution into the tissues and also on its elimination. The high log P value will lead to low effect and increased side effect because the drug will get stuck inside the tissues because of its high lipophilicity. Now we will see what is the Lipinski rule of 5 and what is drug likeness. So you might have heard about the drug likeness. Whenever the new chemical compounds are synthesized, their physicochemical properties are checked to see whether these lead molecules can become the drug. Log P is an important component of the Lipinski's rule of 5. There are 5 general criteria for drug likeness. The criteria involves the parameters like log P, molecular weight, then the chemistry related points. So all these points are given by the Lipinski's rule of 5. Out of that, log P is an important consideration. Log P recommendations are there which predict the drug likeness for new synthetic drug molecules or the chemical compounds. Log P less than 5 is given by the Lipinski's rule of 5. If the log P is less than 5, then it will be moderately lipophilic and it will be suitable for the oral route. Log P value less than 5 and ideally between 1.35 to 1.8 will be the ideal drug for oral formulations or oral route. For central nervous system target drug, the log P value should be around 2. Because for CNS activity or to cross the blood-brain barrier, higher lipophilicity will be required for the drug molecule. For sublingual route drugs, log P value should be above 5. So highest lipophilicity will give you the good candidate for sublingual route. Then what is log D? What is the difference between log P and log D? So log D is also similar to the log P, but while determining the log D, the aqueous phase for the test is replaced with the specific pH media, and that specific pH media may be 7.5 media, and also pH of the aqueous phase in the physiological range can be taken to study the drug molecule absorption behavior in the GI tract pH or the physiological GI pH range of 1.2 to 7.5. You can also select other pH ranges for your drug molecules. Log P, log D, lipophilicity, these are the physicochemical properties of drug which plays great role in the drug development and analysis. So based on the log P, the HPLC analysis is also developed. Now, how to determine the log P? What are the methods? Shake flask method is there, HPLC method is there, and also some softwares are there which can predict the log P values. Log P value is also called as or referred as KOW or POW. Some examples I have enlisted here like paracetamol, simvastatin and acinapine maleate. Paracetamol is having the log P of 0.46. It is BCS class 1. Half-life is 1 to 3 hours. Bioavailability is 7 to 90%. VD is 0.9 liter per kg and protein binding is 15 to 21%. So you can relate these things with each other, like log P is 0.46. BCS class is 1, so it is highly soluble. It is not much lipophilic, that's why its bioavailability is good. It is having good hydrophilic nature, that's why its volume of distribution is not too much, and because of less lipophilicity it is having less protein binding. So I am giving you the examples for your easy understanding. Simvastatin is the another API. It is formulated into oral tablet generally. It is having log P of 4.68. That means it is lipophilic in nature. Its BCS class is 2. Its half-life is 5 hours. It is having low oral bioavailability, that is 5%. So now you relate the bioavailability with log P. Higher log P will give you less bioavailability because this drug will not partition into the aqueous phase. VD is higher. So as lipophilicity is more, VD will be higher. Now protein binding is also higher because of the higher lipophilicity. So these examples are for your easy understanding. You can take other examples also. Now acinapine maleate. These are formulated into this API is formulated into sublingual tablets. Log P is 4.9, BCS 2, half-life is 24 hours. It is having low oral bioavailability of 2%, but the sublingual bioavailability is 35%, and that's why it is formulated into sublingual tablet. It is lipophilic in nature. Its VD is 20 to 25 liter per kg, and it is highly protein bound compound. You can take other examples and you can relate the effect of log P onto the absorption, distribution and elimination of the drugs. So this is small video regarding the log P, log D and its impact and importance in the drug development. Log P is a key physicochemical property of the API which affects its absorption, distribution and elimination. So it affects its bioavailability. Also, it has impact onto the safety and efficacy of the formulation. Also, log P is considered to formulate the drug into suitable formulation. Highly lipophilic molecules can be formulated in the formulations like sublingual formulations, creams, then the soft gelatin capsules, because they are lipophilic and oils are used as base to dissolve these type of APIs. So there are many things which are required to be explored for log P and log D, and I hope this video will guide you to understand the terminologies in a great way. Thank you for watching the video. Please do like, share and subscribe to Pharma Learning in Depth channel.